Good afternoon. My name is Hervé Dombret. I am an hematologist in Paris, in France, and I am the current chairman of the European School of Hematology. The European School of Hematology is a non-for-profit organization. It was created in 1985, and the mission of European School of Hematology is to accelerate and promote the diffusion of knowledge in hematology at the European and now international level. So it's my pleasure today to launch and introduce the first annual John Goldman Award, ESH Award, supported by Novartis Oncology, to encourage research on the treatment-free remission concept. So my first question today is for François-Xavier Mahon. What means treatment-free remission at the clinical level for the patients, and what is the impact of treatment-free remission?
Okay, before I answer the question, I just want to introduce myself. I am François-Xavier Mahon. I am hematologist in Bordeaux, France. I am also in charge of the French group working on CML, chronic myeloid leukemia, this famous disease where effectively we have developed the concept of treatment-free remission. Treatment-free remission is the possibility to stop treatment in patients who are in a very good response. When we have developed the new treatment of CML already 20 years ago, at that time we thought that the treatment will be for all the life, but recently we have demonstrated that it's possible to stop the treatment, and when the patient stops the treatment, half of the patients are still off the treatment long time after the cessation of treatment. The question, and the big question, is that we do not understand why some patients are still off the treatment without a molecular relapse as compared to the patients who have a molecular relapse. So I think that it's very important to continue the research on this disease to better understand this treatment-free remission, which became or which is going to become the most important goal that we need to reach in that disease, because that disease now has a life expectancy which is close to the healthy population.
Thank you very much, François-Xavier. So we all understand that this concept is really important, not only in CML. CML is a model, and certainly the treatment of CML has been and is still a model in hematology. So this concept of treatment for remission is really important for CML patients, but it will become, and it's becoming, more and more important also for other chronic hematological malignancies like CLL or MDS, for instance, but also now for acute diseases like AML or acute lymphoblastic leukemia, because of the maintenance treatment and the duration of maintenance therapy has something to do with the treatment for remission concept. So my second question is for you, Daniela. It's for the biologist, for the researcher. Do you think, what are the main determinant of this concept, treatment for remission? Why some patient can stop the treatment while the other one cannot? Is it only due to immune surveillance, or is it something else?
So first of all, I would also like to introduce myself. My name is Daniela Krause. I'm a research group leader in Frankfurt, Germany, also a laboratory and transfusion medicine specialist. I'm also co-chair of the Chronic Myeloid Leukemia meeting of the European School of Hematology and a member of the Scientific Committee of the European School of Hematology. And so to come back to your question, we think that the concept of treatment-free remission and why it works in some patient populations and not in others might be due, on the one hand, to leukemia cell intrinsic factors as well as extrinsic factors. So intrinsically, there might be the persistence of BCR-ABL as the driving oncogene, or the resistance to certain medications that have been given. But of course, immune surveillance, and we've just heard many talks about this today, could be a very major factor and has been shown to be a major factor in controlling the response or the treatment-free remission incidence. And also, I think there are many components of the bone marrow microenvironment and the surrounding tissues that might be controlling how a patient might respond to the withdrawal of tyrosine kinase inhibitor therapies.
Thank you very much, Daniela. So now I will introduce Ilaria Pagani. Ilaria Pagani is the first winner of this first annual John Goldman Award on treatment for remission. So Ilaria, if you can briefly introduce yourself, where you are working, and briefly summarize your project.
Sure. I'm Ilaria Stefania Pagani from the South Australian Health and Medical Research Institute of Adelaide, Australia. My work is working on TFR and in precision to understand why 50% of the patients undergo to molecular relapse. I would like to thank you, the European School of Hematology, for granting me this award. It's an honor and a privilege receiving this funding and being the first John Goldman awardee. The project for this award is a result of a collaboration with a team of researchers based on summary. So, in summary, with this funding, we will develop a new bioassay, and we will use the most robust bioassay, and we will integrate them to clinical data using artificial intelligence. And this will help in clinical decision making. For example, if a patient is predicted to achieve TFR, we can sure stop TKI in this patient, and without the risk of relapse and without emotional stress. On the contrary, if a patient is predicted to relapse, we can do therapeutic intervention. This means that we can keep maintaining the therapy for this patient and repeat after 1 or 2 years. Or we can intensify TKI therapy, or we can give additional drug like immunomodulators.
Thank you very much.
Thank you so much.
So I have to add that the committee really like the network you had to achieve this goal. This is fascinating, and we hope that the help of the award could help you to produce very interesting results in the couple of years, I hope.
Yeah, thank you so much. Another crucial point for this project is the sample size. Therefore, we are willing to collaborate with international collaborators to increase the sample size and the power of machine learning system.
Okay, thank you.
Thank you so much.
So Rita Ferreira is from Novartis Oncology, supporting the award. So I would like to know, Rita, what is your commitment with ESH on that point and for this year and the future years?
Thank you. So my name is Rita Ferreira. I am currently the medical director for CML in Region Europe in Novartis. And we are really very happy and very proud to be able to keep this partnership with the CML community. As a company, we have been in CML from 20 years now. And honestly, I can tell you that I am very proud to work in a company that didn't settle when imatinib came and changed the life of patients. We kept working, founding new drugs, more potent drugs, and now being able to make a big number of patients free of drug and coming closer to a normal life, I would say. So for us, it's a pleasure. It's a commitment that we have with the European School of Hematology and other societies and work groups in CML that we intend to keep in aiming to support the patients in CML. Therefore, we are very proud to have this award, this first award being delivered this year, and we intend to keep this support with a second award next year during the next 2020 ESH CML meeting.
Thank you very much. Thank you very much for Novartis Oncology support. Thank you, all of you, to have explained today what is this TFR concept. And congratulations for you, and good luck for your next work. Thank you.